Namaste, welcome to Sarat Chandra IAS Academy. So today we'll see the Nobel Prize in Medicine. So this year the Nobel Prize has been declared for two people. One is Victor Ambros, the second one is Gary Ruvkun. So from a UPSC perspective, you need not remember their names, but what we need to understand is their contributions. So how their contribution is helping humanity in advancing its purpose. So what did these people do? So broadly what they have done is they have studied the impact of microRNA and the role of microRNA in gene regulation. So the keywords that we need to understand from this is the role of microRNA and how it impacts gene activity. So we'll understand both of them, but before that, one area where students often get confused is between microRNA and mRNA. So both are not same. So in case of mRNA, so this is called as messenger RNA. This is a different type of RNA compared to microRNA. So microRNA is just a small RNA. So this is not a messenger RNA. This is just a small part of RNA. Now if you see the functional differences, so their functions are entirely different. In case of messenger RNA, the function is simply to convey the code, convey the instructions from the gene for protein making. But in case of small RNA or microRNA, the main function is gene regulation. So student should be very aware about the differences. Do not get confused by the names. mRNA here means messenger RNA, microRNA just means a smaller RNA. So don't get confused. Now we'll get back to the topic. Here these scientists, what they have discovered is today's topic. Okay, so what are microRNAs? As we have discussed just now, microRNAs are nothing but the RNAs which are small, which help in gene regulation. So how do they regulate the gene? First of all, what is the question of gene regulation? So here we have said there is something called as gene regulation. Now the question is, what is gene regulation? So if you observe the human DNA, so we know that human DNA is in the shape of semi-helical arcs. So these are double helix structure. Now this double helix structure, which composes of proteins and amino acids, constitutes our DNA. And our DNA is same in every cell. So all of the body cells contains the same DNA. Now the question is, if all body cells contains the same DNA, how does each cell know how to differentiate its function? For example, the stomach cells, nerve cells, optic nerves, so all of them have different functions. So when they all of them have different functions, how does same DNA will differentiate into multiple functions? So that is where the role of microRNA comes. So what happens is, this microRNA forms a bond with the DNA So what happens? This will form a bond with the DNA and activates. This microRNA activates only selected regions of the gene. So due to this, selected genes will get activated. So when the selected genes get activated, a proper protein synthesis happens. So that is how each organ distinguishes itself from the different organ. So different cells have different microRNA. So when different microRNA synthesizes different proteins, that is how you get different cells. Now we'll come back to their work. So how did they discover this? So we have seen in the previous UPSC questions that they have asked about mRNA vaccines. They have asked about protein synthesis. Now we are going into a step back. So in a normal human cell, what happens is, from the complex DNA, we have functional units which are called as genes. These functional units undergo the process of transcription and translation. So in case of transcription and translation, this mRNA acts as a vehicle. So as we discussed, this is messenger RNA. Now, when this mRNA is undertaking protein synthesis to form different cells. Now, the same question arises, how does it know which cell to form? So that is where the role of microRNA comes. So in microRNA, these scientists have observed that this microRNA forms the link between the protein synthesis from the mRNA into actual protein manufacturing. Now they have studied on an organism called as E. elegans, which is of 1 millimeter size. So just 1 millimeter size, a small organism with just 1 million base pairs. So in its DNA, it has only 1 billion base pairs compared to human genome, which has a billion base pairs. Billion base pairs. They wanted to study something simple to understand its impact. Now, in this, there are three important muscles. One is a nervous system of this organism, then the next one is gut, the next one is muscle. So these three systems are coming from the same DNA. Now, they wanted to understand how this same DNA is getting different muscles. So how this is becoming different cells. So from their research, they have understood that there is an intermediate mechanism called as microRNA. So this microRNA is a new discovery because it helps us understand that there is a intermediate step from this nervous system from DNA into mRNA. So this step is a new discovery for which the Nobel Prize has been given. Now what is the significance? So the significance is very simple. By understanding the role of microRNA and how it is blocking and enabling the protein synthesis, we will be able to make the new medicines. So this will help in biotechnology research, this will help in medicine development. This will also help in development of precision medicine. So precision medicine is a medicine which is customized for a single person. So based on my own DNA, each person has a separate DNA. From that, we will try to synthesize a medicine suitable for that person alone. So this is the role of microRNA in synthesizing the mRNA and in synthesizing the protein for different cells. Thank you.